a way to get people's attention. All right, thank you everyone for coming today to Lauren Parker's thesis defense. Yes, I'd like to thank everyone who's joining us online. For people online, if you have questions after the defense, put them in the chat and we will read them out for Lauren. I'd like to thank her family for coming today. Her mom and her dad came, her sister, her partner, and some of his family and some other friends, so thank you. And the rest of her thesis committee. So her co-advisor at CSU Monterey Bay, James Lindholm, Gita McDonald here at Moss Landing, and Andrew DeVogelaire at the Monterey Bay National Marine Sanctuary. Right, so a little bit about Lauren before she gets up here and gives her presentation. So you may be wondering, what does the M stand for? And I don't have a copy of her birth certificate, but I presume the M is for mermaid. You see, she is more aquatic, really, than terrestrial here, even trying both things. Maybe not so successfully. But there's a lot of evidence growing up that she spent a lot of time in the water. That's where it all began. In pools, even, I think here she was studying for a final exam. Couldn't leave the water. And doing very mermaid-like things. Much more at home, I think, in the ocean than on land. Also, like a mermaid, she can communicate with lots of different creatures, both marine ones, sea stars, fish, sharks, baby sea stars, and those animals on land. Cats and dogs, donkeys, parrots, more dogs, lizards. She's also very adept at communicating without using words. She can make some good faces. She's probably making one at me right now. Here, she's either showing her displeasure about having to cook these crabs, or it's a devilish grin, like she can't wait to put them in the boiling water. Here's another look. Maybe that was after eating the crab. I don't know. Over 20 years or so, she perfected that look. Again, I think that look is coming at me right now. But she also has a very infectious smile. She's got the silly face, and whatever this one is. Right, Lauren's really at home, out in nature, communing with nature, climbing trees, exploring the coast, just hanging out with a bunch of foliage, right? Being one with the desertness of Baja, right? In the water, above the water, near the water. Lauren's really at home right in nature and has really developed a really strong appreciation for ecology, for spending a lot of time making observations. She's also a compassionate and patient teacher, and this has been a big part of what she's done. So she was a TA for our subtitled ecology class the year after COVID. Down in Santa Barbara, she was a marine science instructor with the junior lifeguards. She's been a docent for many years with the Hollister Ranch Tide Pool School with K-12 kids, getting them excited about marine science, and as well teaching kids how to swim, like you can see in this photo here. In this one, I think she's teaching her sister how to pretend to be excited to be standing next to a bunch of dead fish. And here, when we were down in the Baja class, she realized this was an opportunity to teach some of us how to do a better job at drinking carbonated beverages. And here she was demonstrating to me how to do it, and then me as the pupil perfecting said technique. So good teaching job. Thank you. Right, Lauren did her undergraduate degree at UC Santa Barbara, and while she was there, she spent quite a bit of time underwater, blowing bubbles, learning how to scuba dive. So she was a lab assistant and a technician with PSCO, the Partnership for Interdisciplinary Studies of Coastal Oceans. There she did a lot of scuba surveys in kelp forests, so she gained a lot of expertise. How do you run a big monitoring program? How do you do ecological surveys? A lot of knowledge on algae, invertebrates, and fishes, understanding what's happening inside and outside of marine protected areas. She was also a lab assistant with the Santa Barbara Long-Term Ecological Research Project with a grad student there who was studying invasive sargassum in Catalina. So she came here to us having a really deep background, scuba diving and doing ecological surveys. Right, and so she came to Moss Landing to work with me and to work with James Lindholm at CSUMB to really master the art of counting things. And she got involved with James' projects that was doing these deep water sort of stereo video scuba surveys of what was happening at the head of the Carmel Canyon at a really sort of high temporal intensity. And so that's what she'll be telling you about for her thesis today, understanding seasonal changes and sort of habitat affinities of fishes on some of these deeper reefs. Right, some other things while she was here was she was a fellow with the NOAA CCME program. Part of that was doing an internship, and so she did that at the Monterey Bay National Marine Sanctuary. That's how she got to work with Andrew de Vogelaar. And there she was doing a bunch of various kelp forest video analysis, kind of related to a lot of the very time-consuming video analysis she did for her thesis. She got to participate on an expedition after the Davidson Seamount to use ROVs to study what was happening on the top of these big underwater mountains. She presented a number of times, both at the Western Society of Naturalists and a NOAA West Coast Biological Observations Workshop, and she received a number of awards, twice the Moss Landing Scholar Award. You know, Lauren's been on a bit of a circuitous journey, you know, to get here. And partly I think that's because she really had this bug for international travel. And so a lot of times I was like, where in the world is Lauren Parker? And I think some of this came from when she was younger, and I think she would sleep in a suitcase. And that haircut, by the way, no notes, perfect. And so part of it in 2017, this was my fault. I sent her on with some colleagues on an expedition to Tonga to help do some surveys of coral reefs in nice warm tropical waters. In 2018, again, counting things, the theme that comes through. She went on this to count things, right? She was on the 2018 Baja class trip to Isla Natividad. There she did an independent project where she was, again, counting things and counting fish, but trying to help the fishermen on the island estimate what was the value of their fisheries resources inside and outside of the MPAs. So knowing what fish were there and kind of what the value was, like how much could they get? So again, theme, counting things. 2019, she went on the Global Kelp Systems class trip with me and Mike Graham to Chile. Again, more mermaid stuff, you know, more counting things, comparing differences in ecological communities inside and outside of kelp forests. And they're making another wonderful face. That was where this little tiny truck had all of our dive gear and could not make it back up a very steep slope. We made a big traffic jam. Again, counting things. Lots of other trips. Many of these with her sister. Italy, Peru, I think Cuba. I had to look up some of these. I was like, is that really? They let you go to Cuba? Crazy. France, Germany. Even when she was racing to get me a copy of her thesis so that she could run off to Paris because she had to see Taylor Swift. So, you know, Netherlands. But sometimes she stayed close to home. So for a number of years, Lauren was the lead technician for me running our surf zone marine protected area monitoring project. So taking a lot of the skills that she had learned from these other monitoring programs, and she was the one that planned all the logistics, all the organization, you know, leading the field work, dealing with the data, managing all the people, and she's really fantastic at all of these things. You know, sometimes they would catch really big fish, and they would have to drag those to shore. Again, she's very good at counting things. She was also a co-author on a recent publication that we had that was evaluating the effects of MPAs in the surf zone. And then maybe 2 years ago now, she moved back to Santa Barbara, and she's been working with Jenny Dugan, who's the lead statewide for this project, and so she's the lead technician now helping to run that surf zone project there. And so with that, I'm going to hand it over to Lauren. I think this is probably her final message to me after this introduction, is that. And so, Lauren, please tell us about your thesis.
Okay, thank you, Scott, for that introduction. And thank you all for coming today. So I am here to present my master's thesis work titled Seasonality of Groundfish Community Structure and Habitat Associations on a Mesophotic Reef in Monterey Bay National Marine Sanctuary. Here's a quick outline for this talk. First, I will begin with an introduction to the project and the system that I studied. Then we'll talk about some objectives of the study. We'll talk about the methods that I used to complete this work. We'll get into some of my results. And finally, we will talk about some conclusions. All right, here we go. The arrows at the top of the screen will act as a kind of a guide through this talk. So as we move through each section of the talk, the corresponding arrow will be highlighted. First, I'll begin talking about the subtidal community. Communities are complex systems that can be characterized by a variety of factors. Their structure is heavily dependent on habitat availability, as well as the kinds and numbers of species present. They can also be characterized by their dynamics, or how that community changes over time, and in response to local environmental variables. These important factors that determine community assemblage then influence patterns of reproduction and feeding dynamics like prey abundance and prey availability. So understanding the dynamics of a subtidal community structure is critical to effective management of marine systems. The idea of ecosystem-based management is an integrated approach that incorporates the entire ecosystem into resource management rather than just a single species or resource. So it takes into consideration the effect of physical habitat and abiotic environmental factors on species dynamics, trophic interactions, and the relationship between marine systems and human activities. This highlights the need for continued characterization studies of marine systems that will include a comprehensive study of these factors, habitat, environmental drivers, and species information in order to update and inform management. Determining the connection between ocean climate variability and marine population dynamics is essential to understanding a community's ecology in terms of things like food availability, reproductive success, and survival thresholds, particularly in the face of a changing climate. The image on the top is an image plotting global sea surface temperature anomaly over the last century, showing a steady increase. And the bottom image shows synchrony, or these kind of concurrent fluctuations over time between biological processes such as chlorophyll and primary production, and physical processes such as sea surface temperature and currents. Changes in environmental variables are often the result of shifting water masses due to surface wind patterns. The wind-driven upward movement through the water column is known as upwelling, a common phenomenon in many coastal regions of the world. Increased upwelling conditions occur when characteristic wind patterns combined with the Earth's rotation move surface waters offshore, allowing for colder, nutrient-rich water from the deep ocean to be pulled up or upwelled to the surface. And then these surface waters experience much higher light conditions, which then support photosynthesis. As upwelling depends on characteristic wind patterns, these movements of water masses are often seasonal. Upwelling conditions are more prevalent in the spring in the Central Coast when northerly winds, or winds that blow from the north, blow parallel to the shore and drive pulses of cold, nutrient-rich water that are often low pH and low in oxygen close to shore. This influx of nutrients to the surface waters influences primary production via photosynthesis. This means that all the teeny tiny algae floating on the surface are given a bunch of food all of a sudden, and they use that to proliferate. The increase in primary production creates a food source for zooplankton and other lower level consumers like these bait fish, which in turn feed higher and higher trophic levels. And then some of these things feed each other. It's very complicated. This is a general food web. Another important aspect of community structure is the availability of suitable habitat. A habitat's the physical space that an organism occupies, provides it with resources, conditions, and space that it needs to survive. The idea of landscape ecology considers the structure and function of landscapes, or seascapes in this case, in conjunction with everything we've talked about already, like environmental factors, to understand the spatial distribution of species or communities. Essentially, the physical space that an organism occupies is as important in its surrounding environmental conditions. So this brings us to the idea of habitat selectivity and the idea that seasonal patterns of community assemblage are also a function of specific habitat characteristics of the benthos. When talking about habitat associations, however, the scale of study is important. At finer scales, fish may associate with habitat features like a coral head, a sand trough, a rocky reef outcrop, while at larger scales, they may associate with certain depths or even a habitat feature like a submarine canyon or a seamount. Therefore, when talking about habitats and subtidal habitats in particular, we can divide them into different categories. We have megahabitats, which are on the scale of kilometers to tens of kilometers and would be something like an entire continental shelf. Mesohabitats which are on the scale of tens of meters to a kilometer and might be something like a small seamount or a canyon. This is the Carmel Canyon where we did this study, if you can see it at all. Macrohabitats, which are on a scale of 1 to 10 meters, such as large boulders or reef structures. And finally, microhabitats, which are typically less than a meter and include sand, pebbles, small cracks, and crevices. This rosy rockfish hiding in a crevice. For the purposes of this study, I'll be focusing on these two, macrohabitats and microhabitats. Now, microhabitats are made up of microhabitats, and the high level of fine-scale variability within them is itself a known driver of species distribution. In particular, I'm considering the microhabitat attributes of relief, or vertical steepness of the habitat, rugosity, or how complex the structure is, how many holes and crevices there are, and substratum type, what was actually on the bottom, whether that be hard like bedrock or soft like sand. So let's meet some of the species we'll be talking about. Pacific coast groundfish are a grouping of several different species, but all of them live on or near the ocean floor, and generally include rockfish, roundfish, flatfish, and several species of sharks and skates. There are quite a lot of them. These images are missing many of the species, including all the flatfish, sharks, and skates, but you get the idea. There are a lot of them. So talking about a few of the major players in this study, rockfish there's 102 known species, 65 of which are primarily distributed along the Pacific coast of the United States. Rockfish are highly variable in terms of everything, feeding mode, size, structure, habitat associations, depth preferences, everything. Rockfish reproduction cycles are highly seasonal, and many species exhibit something called ontogenetic shifts in habitat and depth use. So rockfish of different age classes occupy different habitats and depth profiles. Rockfish have a pelagic larval phase, which is followed by settlement to nearshore reefs several weeks to months later. Some of the other major groundfish players are the greenlings, lingcod, and kelp greenling. Both species also have a pelagic larval phase, followed by settlement to the nearshore environment. These species also display ontogenetic shifts in habitat use. Both species exhibit nest selection and guarding behavior, where males will select and guard optimal nesting areas before the arrival of reproductive females. Lingcod are also cannibalistic, with the adults often consuming younger individuals. So why mesophotic reefs? These kinds of characterization studies are relatively well documented at shallower depths, with surveys often conducted on scuba. And several studies exist for deeper depths, which are often studied with the use of submersibles, such as ROVs. However, studies in this middle zone, the mesophotic, are relatively rare, leading to a gap in the data available to inform management. So let's talk a little bit about the objectives of my study. This study focused on the characterization of community structure in terms of population abundance, biomass, and diversity of groundfish on a mesophotic reef in the Carmel submarine canyon. I looked at the influence of seasonality and depth on community structure. I then looked at the influence of local environmental variables on community structure. And finally, I examined key species selectivity of microhabitat categories and tried to interpret these as habitat preferences. Get into my methods. Some water first. Okay. To start off, I'll talk a little bit about my study site. This is a canyon system just offshore the Monterey Bay called the Ascension Monterey Canyon System. This is a canyon system with 6 canyon heads, 3 up in the northern part, called the Ascension, Año Nuevo, and Cabrillo canyons. The southern part made up of this giant canyon, the Monterey Canyon, with the Soquel Canyon off the top of the Monterey Canyon, and then down here at the bottom we have the Carmel Canyon. Now if we zoom in a little on the Carmel Canyon, it approaches very near to shore at a place called Monterey Beach, which is a dive site most people here are familiar with. This is a unique site in that the northern head of the Carmel Canyon comes very near to shore and right up to the edge of the kelp forest, making it easily accessible via scuba, which can also lead to the large waves that can sometimes be present at this site, and the phenomenon for divers known as the monastery crawl, which is what it sounds like, it's crawling out of the water, trying to time the waves. Surveys for this project were conducted on scuba from March through December of 2017. January and February surveys were not possible due to weather conditions. A diver-operated stereo video camera was operated horizontally along a permanent transect at 40 meters depth, vertically up the wall from 40 to 30 meters, and then horizontally along the top of the wall at 30 meters, just like in this little diagram. The survey design was to facilitate continuous data collection with a single team on a single dive while looking at multiple depths. Slide for the camera. This is a stereo video camera used to collect data. Stereo video camera setups are dual lens. This one consists of two cameras inside houses, bolted to either end of a 1 meter aluminum bar, allowing for a fixed camera angle and an overlapping field of view. The camera setup also had dual strobes to illuminate the steady site, and a foam lining inside the aluminum bar helps to keep the rig almost neutrally buoyant. The next step was to collect information from the videos. This was done using the video processing software EventMeasure. EventMeasure was formulated specifically for collecting biological data from digital imagery. This is what the data collection window looks like. As you can see, both camera files are loaded into the software. The videos are synced using a corresponding moment in both videos, and they are able to be analyzed in tandem. Each fish can then be identified to species and measured by clicking the nose and the tail of the fish in both frames. The program then gives an accurate length measurement of each individual fish. Species information is selected from this little drop-down window that pops up when a length measurement is completed, and this little 3D information box is used to evaluate the quality of each measurement, which just means that it lets me know if I need to redo the measurement and I've collected points that are wildly outside of the norm. Sometimes the transects aren't too fishy, like the top image, but other times there are quite a few fish, like the bottom. Got a lot fishier than this. This was just the best quality image that I could find with a ton of fish in it. You can also see there was differences in light availability down there, depending on the day. In order to look at habitat as a predictor of fish assemblage, I collected habitat information from the videos as well. This was done by taking multiple passes through the video, looking specifically at habitat. In the first pass through the video, I divided the transect into sections divided by obvious changes in substratum type. So this is obvious changes in the benthos or the bottom, and these changes were marked using timestamps. The categories were hard if greater than 70% of the benthos was bedrock or boulder, mixed hard if 50% of the habitat was bedrock or boulder, and about 20% of the remaining habitat was cobble or small rocks, mixed soft if 50% of the habitat was soft sediment such as sand, and about 20% of the remaining habitat was cobble or small rocks, and the substratum was considered to be soft if greater than 70% was soft sediment like sand or mud. The second pass through the transects was used to collect relief and rugosity information. Relief is again that vertical relief or steepness of the habitat, and rugosity is a measure of habitat complexity. These categories were estimated for the approximate 1 square meter surrounding each fish, resulting in a substratum relief and rugosity measurement for each fish. Relief was considered low if less than 25 centimeters, medium if between 25 and 100 centimeters, and high if greater than 100 centimeters. Similarly, rugosity was considered to be low if the habitat was flat with no holes or crevices, medium if small holes and crevices less than 25 centimeters were present, and high if there were large holes and crevices greater than 25 centimeters for fish to hide in. A final pass through the videos was used to collect uniform point contact information, or UPC. This was done to collect habitat information independent of fish presence. A measurement of these habitat characteristics was collected for every 10 seconds of elapsed video time for each transect on a subset of dates, using the same methods I just described, but using the center point of the video instead of a fish every 10 seconds. And finally, those microhabitat categories were combined into a single habitat variable labeled as substrate relief rugosity for both the habitat used by each species and the UPC habitat collected. As a last point, these habitat variables were then used to calculate a selectivity index called Manley's alpha for each species, which I'll talk about a little bit later. To look at environmental variables as drivers of fish community, the variables of temperature, pH, dissolved oxygen, and salinity were collected using a CPOX sensor at South Monastery Beach. This sensor was collecting oceanographic information for the Croker Lab at UC Santa Cruz, and they graciously lent me their data. These data were measured every 30 minutes, and I averaged them to a single value for each variable for each survey date. I downloaded localized upwelling indices, the Coastal Upwelling and Transport Index, or CUTI, which measures nearshore vertical transport, or upwelling and downwelling, and the Biologically Effective Upwelling and Transport Index, or BEAUTY, which measures nitrate flux. Nitrate is important as a key nutrient brought to the surface during upwelling that drives productivity. These indices were measured daily and reported in 1-degree latitudinal bins. In order to run the analyses on this data that I wanted to, there was a significant amount of data preparation. I calculated abundance as the total number of species present on each transect, and density as the abundance divided by an approximate transect area of 1 square meter. Biomass is measured as weight per unit area and was calculated using the fish length information painstakingly collected from video analysis and commonly accepted length to weight coefficients that are unique to each individual fish species. I calculated Shannon diversity as a single value for each survey date and using my abundance data. And then I calculated species richness and evenness values for each sampling date based on those Shannon diversity values, as richness and evenness are components of diversity. Each survey date was also assigned a month and a season for further analysis. Now, on to some data analysis. In order to look at seasonal and depth-related differences in community structure, I performed a two-way analysis of variance, or ANOVA, on my total density, biomass, and diversity data using season and transect as a proxy for depth, as my factors. To dig a little deeper into the specifics of the community, I used a multivariate approach and ran a non-metric multidimensional scaling analysis, or an NMDS, which is an ordination technique that allows you to view multivariate data, but it doesn't allow you to test for significance. For that, I used a permutational analysis of variance, a PermANOVA, and a similarity percentage analysis, or SIMPER, to look at the contribution of individual species to the dissimilarity between groups. And again, those groups were season and transect. These multivariate tests were run on density and biomass data only because there was no significant difference in diversity, seasonally, or among transects. It's also worth noting that these data were log transformed prior to analysis to downweight the importance of particularly abundant species. To look at the effect of environmental variables on community composition, I used a distance-based redundancy analysis, or DBRDA. This test essentially takes a series of explanatory variables, which in this case were the local environmental variables collected with the sensor, and tries to relate them to a series of response variables, which in this case were my density and biomass data. And finally, to look at habitat associations, I used that metric called Manley's alpha, which is a parameter used to measure preference for a commodity compared to the availability of that commodity. And it's typically used to look at prey selectivity, but the variables used to calculate it can apply to habitat as well. So I used it to look at species selectivity for or against specific microhabitat categories available on the site. Now, let's get into some results. Overall, 2,315 species were counted, measured, and identified. These are a few of the species seen on my transects. The majority of them were rockfish species, like these fish here. This is a vermilion rockfish, a starry rockfish, and a rosy rockfish. There were also several other larger-bodied species, like this lingcod. And then the two on the end are also rockfish species. We have square spot and half-banded rockfish, which were the most abundant species on the site by far. So, when looking at our seasonal and depth-related changes, in terms of total fish density, these are some plots to show the difference in density between seasons and between transects, which again were used as a proxy for depth. To orient you to these plots, the y-axis is mean density, measured in fish abundance per square meter. In the plot on the left, the x-axis is season, and in the plot on the right, the x-axis is the different seasons grouped by the depth transects, labeled as the 40-meter transect, the vertical transect, and the 30-meter transect. The bars themselves represent the total fish density, and the error bars here represent standard error. As you can see, density differed significantly between the seasons. Spring had the lowest total fish density compared to fall and winter. There was no significant difference between the different depths, and the interaction factor between season and depth was not significant, indicating that the seasonal patterns are consistent between the different depth transects. These plots show the difference in biomass between season and between transects. Similar to the density plots, the y-axis is the mean biomass measured in kilograms per square meter. In the plot on the left, the x-axis is season, and in the plot on the right, the x-axis is the different seasons grouped by the depth transects. The bars, again, represent total fish biomass, and the error bars here are also standard error. Again, similar to density, biomass differed significantly between the seasons. Summer had the lowest total fish density, biomass, compared to the fall and the winter. There was no significant difference in biomass between the different depths, and the interaction factor between season and depth was not significant. Now on to diversity. These plots show Shannon diversity on the top, richness on the bottom left, and evenness on the bottom right among seasons grouped by the different transects. Diversity, richness, and evenness are on the y-axis, and the different transects are on the x-axis. The different colored bars represent the different seasons, and the error bars are standard error. There was no difference in species diversity or species evenness among seasons or depths. There was significantly lower richness on the shallower 30-meter depth than both the 40-meter and the vertical transect. There was no difference in richness among seasons. So, getting into a little bit of the multivariate stats. These two figures represent the results of the non-metric multidimensional scaling analysis, or NMDS for density. Both of these images have the same data in them, the density data represented by the points in the image and the overlaid species vectors. An NMDS is a representation of similarity in a multivariate space. So the closer the points are together, the more similar they are, and the further away they are, the more dissimilar they are. The difference between these plots is in the groupings. The image on the left is grouped by season. The image on the right is grouped by the different transects represented by the different colored ellipses. I'll walk through these images a bit more, but in order to interpret them, the direction of the species vector, or the arrows in the image, with the ellipse indicates a positive relationship, and the length of the arrow represents the strength of that relationship. So the longer the arrow, the stronger the relationship. Now, remember, the image won't give us any statistical significance, so I ran a Permanova and found that fish community density differed significantly both among seasons and depths. These plots show the same density data, but with only those vectors that have significant positive relationship with the different groupings. This simplifies the plots a lot more, and we're able to discern some of the species that are driving the seasonal and depth-related differences in density. So in this plot, we can see that kelp greenling have the highest densities in the spring and summer, and on the vertical transect in the shallower 30-meter transect. Square spot and half-banded rockfish are present in the summer, fall, and winter, and seem to have high densities on all three depth transects. In order to look a little deeper into how much each species is contributing to the differences in community density between seasons and depth, I ran a similarity percentage analysis, or SIMPER analysis. And I found the same 5 species were driving the differences in community structure between both seasons and depths: the half-banded, square spot, rosy, blue, and olive yellowtail rockfish. Greater than 50% of the variability in density between seasons was driven by half-banded and square spot rockfish. The greatest seasonal difference in community assemblage was between winter and spring, which, if you remember, were the ellipses that had the most separation on that NMDS plot. All of the driving species were more common in winter compared to spring. And the greatest difference in community density between depths was between the deeper 40-meter transect and the shallower 30-meter transect. Half-banded, square spot, and olive yellowtail rockfish were more common deeper, and rosy rockfish and blue rockfish were more common shallower 30 meters. Now, if we look at community biomass data, this is another set of NMDS figures, this time for biomass. Again, these images both have the same biomass data in them. The figure on the left is grouped by season, and the one on the right is grouped by the depth transects. The vectors displayed on this plot are only those vectors with significant correlations to season and transects. Looking at the Permanova, I found that fish community biomass differed significantly among seasons and among the depth transects. The interaction factor was also significant, which indicates that seasonal changes were not consistent among the different depths. Now, if we look a little closer at these plots, we can see that the different species group in terms of their biomass. As you can see, these plots have even more overlap than the density plots. But if we take a look at the lower left corner of both plots, we can see that lingcod line up with that purple ellipse and the blue ellipse, indicating that they seem to contribute more biomass in the winter and on the deeper 40-meter transect. This next group rosy rockfish, and square spot rockfish contribute more biomass in the fall and winter and on the vertical transect. And finally, taking a look at this lower left corner, right corner, kelp greenling, gopher rockfish, and vermilion rockfish all contribute the most to biomass in the winter and spring, and primarily on the shallower 30-meter transect. Blue rockfish are a little less selective and contribute similar biomass in all seasons, but specifically on the shallower 30-meter transect. And looking at the results of the SIMPER for biomass, a greater number of species contributed to the difference in biomass between seasons and transects. The biggest contributor to the difference in biomass was lingcod. They contributed 20 to 30% of the difference between seasons and transects. The greatest seasonal difference in biomass between seasons was between winter and summer, where winter had the highest biomass and summer had the lowest. And the greatest difference between the transects was between the vertical transect and the shallower 30-meter transect. With the exception of lingcod, most of these species contribute relatively evenly to the differences in seasons and transects, and the majority of them were present in the winter compared to summer. The increased biomass on the 30-meter transect compared to the vertical transect was primarily driven by vermilion, gopher, and blue rockfish. Okay, so looking at the effects of environmental variables on community composition. These are the results of the distance-based redundancy analysis, or DBRDA, using density data. The points on both of these plots are the same. They represent density data, which is colored by the different seasons, and the size of the point represents the density, so the bigger the point, the greater the density. The vectors on the plot on the left represent the different species, and the vectors on the plot on the right represent the different environmental variables that will run through the model. Now we can interpret these plots by sort of mentally overlaying the two images and looking at the vectors. So if a species vector points in the same direction as an environmental vector, then those two have a positive relationship. Conversely, if the species vector points in the opposite direction of an environmental vector, they have a negative relationship. I found that the environmental variables tested were associated with variation in community assemblage in terms of density. And more specifically, that pH, temperature, and the beauty index, which, if you remember, is that measure of nitrate and upwelling, were significant predictors of variation in community density. These are the same exact plots, same vectors, same significance. Now the points are colored according to the different transects. This gets a little complicated, but if you look at all of these plots together to get a better picture of the seasonal and environmental dynamics of the fish community in terms of density. Now that we've seen all the complicated plots, here's a slightly simpler breakdown of what they mean. In terms of temperature, species that were positively associated with warmer temperatures included half-banded, square spot, rosy, blue, canary, and olive yellowtail rockfish, along with lingcod. Species that were associated with cooler temperatures were kelp greenling, vermilion rockfish, kelp rockfish, and cabezon. When looking at pH, the species positively associated with pH are those same species associated with the lower temperatures, that kelp greenling, vermilion rockfish, kelp rockfish, and cabezon. And conversely, the same species positively associated with an increase in temperature are associated with a decrease in pH. That's the half-banded, square spot, rosy, blue, canary, and olive yellowtail rockfish, along with lingcod. When looking at the beauty index, we have the same results as pH. Same species are positively associated with an increase in pH as an increase in the beauty index. And the same species negatively correlated with an increase in pH are negatively correlated with an increase in upwelling conditions. Since the same species seem to associate with a particular set of environmental conditions, we can sort of group them together and look at the conditions as a whole. In doing this, we can say that the species here on the left, that I won't name again, are present in conditions of increased temperatures, low pH, and low upwelling. All conditions often associated with waters in the summer, fall, and winter. And when looking at the DBRDA plot colored by transect, we can see that these same species are more present in these conditions on the deeper 40-meter transect and the vertical transect. Kelp greenling, vermilion rockfish, kelp rockfish, and cabezon are all more present in conditions of decreased temperatures, increased pH, and greater upwelling conditions. All conditions often associated with waters in the spring, and these species were more often found on the shallower 30-meter transect and the vertical transect. These are the DBRDA plots for community assemblage in terms of biomass. However, When looking at the statistical results, I found the environmental variables tested were not associated with community assemblage in terms of biomass, which just means we cannot attribute changes in the community to those environmental variables in terms of biomass. Now let's take a look at some of the microhabitat associations. In order to look at habitat associations, I looked at the habitat selectivity for those top 5 species from the SIMPR that drove differences in community density between seasons and between depth transects. Half-banded rockfish, square spot rockfish, rosy rockfish, olive yellowtail rockfish, and blue rockfish, and for the single species that drove differences in biomass between seasons and transects, which were lingcod. This is the habitat selectivity plot for half-banded rockfish. In this plot, the habitat categories are on the x-axis, and the selectivity index is on the y-axis. This is the Manley's alpha value that I calculated using the microhabitat variables collected from video footage. Now, remember, the habitat categories are a combination of substrate, relief, and rugosity. The dashed line across the plot represents the null index, and this value represents the likelihood that all available microhabitat categories on the site were selected for equally. If a species alpha score, which are these bars, for a particular microhabitat category is above the dashed line, then we can say that that species selects for that habitat. And if the alpha score is below the null index, we can say that that species selects against that microhabitat category. So from this plot, we can say that half-banded rockfish select for mixed substrate, medium to high relief, medium to high rugosity. I also looked at habitat selectivity by transect and discovered that species habitat selection was more specific to the different transects. These plots are the same selectivity plots, however, now the individual bars are colored by transect, and each transect has its own null index or dashed line. So here we can see that half-banded rockfish selected for mixed substrate, high and medium relief, and high and medium rugosity on the deeper 40-meter transect, did not select for any habitat types on the vertical transect, and selected for mixed hard substrate, high relief, and high rugosity on the shallower 30-meter transect. So going forward, I will show both plots for each of these driving species, but we will just talk about the habitat selection at the different depths. Here we can see that square spot rockfish select for mixed substrate, high relief, and high rugosity on the deeper 40-meter transect, did not select for any habitat on the vertical transect, and mixed substrate, higher medium rugosity, higher medium relief, and higher medium rugosity on the shallower 30-meter transect. When looking at blue rockfish, we can see they selected for hard, and mixed soft substrate, high to medium relief, and high to medium rugosity, but only on the shallower 30-meter transect. They did not select for any microhabitat categories on either of the others. And looking at olive yellowtail rockfish, they did not select for any microhabitat categories on the deeper 40-meter transect or the vertical transect, but they do select for hard substrate, high relief, and high rugosity on the shallower 30-meter transect. And looking at rosy rockfish, we can see that they select for the widest range of microhabitat, and notably on all of the transects. They select for mixed substrate, medium relief, medium rugosity on the deeper 40-meter transect, hard substrate, medium relief, and high to medium rugosity on the vertical transect, and hard substrate, medium relief, and medium rugosity on the shallower 30-meter transect. And finally, when looking at lingcod, lingcod selected for mixed substrate, high to medium relief, and high to medium rugosity on the deeper 40-meter transect, hard substrate, high relief, and medium rugosity on the shallower 30-meter transect, and did not select for any habitat on the vertical transect. I know that was a lot of information, so here's a quick recap of all of those many, many plots and statistics. We looked at temporal and depth-related differences in community structure and found that overall density and biomass differed seasonally. Overall richness was the lowest on the shallower 30-meter transect. Season and transect were both influential in terms of community density and biomass. The same 5 species drove community differences in density between seasons and transects, and lingcod were the major driving species in community differences in terms of biomass. For the effect of environmental variables, we found the variables tested were associated with variation in community assemblage in terms of density, but not in terms of biomass. Microhabitat associations were a bit more complicated to summarize, but in general, microhabitat selectivity was different between the different transects and seemed to vary with available habitat. So let's talk about some conclusions. So my results showed some significant differences, both seasonally and among the different transects. Seasonal differences in fish communities are often the result of reproductive trends, and these trends are linked to changes in environmental conditions. For many groundfish species, including rockfish, mating and fertilization occur in the fall and winter. Larvae are then released in the late winter or early spring when productivity and larval food availability is highest due to increased upwelling conditions. The pelagic larvae inhabit coastal waters before transforming into pelagic juveniles and settling to nearshore reefs several weeks to months later. There's some nuance to this schedule. There are winter and spring spawning species. Some species have multiple broods, but this is a general cycle. Greenlings, which if you remember are also groundfish species, can include kelp greenling and lingcod, often exhibit nest selection and guarding behavior, where male greenlings select and guard for optimal nesting areas throughout the winter before the arrival of females in the spring. So when looking at the spring on my site, we saw both low density and low biomass compared to the other seasons, but that doesn't mean there weren't any species present. Vermilion rockfish, gopher rockfish, and kelp greenling were all present in the spring. I think these species were all likely using the site for reproduction. The majority of fish observed during this time were adults, and this timing matches relatively well for each of these species' larval release periods. These species are also what you might call shallow shelf associating species, or those species that tend to inhabit the deep photic zone, which you might understand to be the deeper part of where light reaches, which could explain their dominant presence on the shallower 30-meter transect, which was immediately adjacent to a kelp forest. And as a final note, greenlings are often cannibalistic, and smaller kelp greenling are often the prey of adult lingcod, which could add to the separation of these two species between the shallower 30-meter transect and the deeper 40-meter transect. When looking at summer, we see a slight increase in density but very low biomass, indicating an influx of smaller species to the site. This is where we see those half-banded and square-spot rockfish start to show up along with rosy rockfish. Square-spot and half-banded rockfish in particular are more associated with mid-shelf habitats, and those habitats in the upper mesophotic zone. This is the area below the photic zone and usually begins around 30 meters depth. Adults of these species could be releasing larvae in deeper waters, which then follow upwelled water in these massive schools up into the depths of this study, where they settle in the summer and the fall. Their association with the deeper waters could help explain their presence on the deeper transects, and may give some evidence that this study area acts as a kind of a transition zone between shallower and deeper associating species. When looking at the fall community, we see a marked increase in both density and biomass, indicating remaining schools of those smaller bodied species, coupled with an increase in larger bodied species, including lingcod, as well as schools of blue rockfish and olive yellowtail rockfish. Blue and olive yellowtail rockfish are shallow shelf and often kelp forest associated species. Blue rockfish in particular are planktivores that often show up in these large feeding aggregations in the summer and the fall months when upwelling conditions have relaxed and plankton-rich waters move shoreward in the absence of driving winds. Schools of this species help to explain the high density and biomass but low richness on the shallower 30-meter transect in the fall. And finally, the winter community also had high density and biomass, largely due to the presence of the same community as the fall. Our schools of smaller half-banded and square-spot rockfish remain, along with those larger-bodied rockfish species and a notable increase in lingcod, accounting for the increase in biomass. I think this increase in lingcod presence could be the result of the same nest selection and guarding behavior as kelp greenling, where males come up from the deeper depths to select optimal nest, which could also help explain their almost exclusive presence on the deeper 40-meter transect. Taken together, here's a little outline of the seasonal community. So let's talk a little bit about habitat. Availability of suitable habitat is an important predictor of the spatial distribution of groundfish species. This site is a very unique habitat. The complex rocky foundation of the head of the Carmel Canyon is in close proximity both to the deep waters of the canyon and to a kelp forest, allowing for a nutrient-rich and highly diverse environment for species from the near shore all the way to mesophotic depths. And because the site is so unique, it will likely to be difficult to extrapolate from these results. Despite the site's location at the head of a canyon, there was quite a bit of variability in microhabitat, and every category of habitat was present. However, the overall dominant microhabitats were hard or mixed hard substrate, higher medium relief, and higher medium rugosity. Almost all species that drove differences in community density and biomass selected for these highly available microhabitats, which was not surprising, as most of them are rockfish species whose habitat preferences are often hard, complex substrata, particularly as adults. Interestingly, microhabitat selection changed or was more specific when looking at the different transects. The different transects had different amounts of each microhabitat available, but were also situated at discretely different depths, not counting the vertical transect, and most species did not select for microhabitat on that transect. This gives some evidence that species are displaying some adaptive habitat usage, in which species rely on both the physical habitat around them, but also on the conditions in which that habitat exists. For example, lingcod selected for habitat on both the shallower 30-meter transect and the deeper 40-meter transect, but were overwhelmingly present on the deeper 40-meter transect. Their preferred habitat of hard to mixed hard substrate and high relief was more readily available on the deeper transect. But their association with highly productive deeper waters also means this transect likely provided more optimal nesting habitat and prey availability for this species. Blue rockfish were the only species that selected for habitat categories outside the norm for their species. I think this is because blue rockfish are pelagic species that are often found in large schools over variably complex substrate and kelp forest habitat. This species was overwhelmingly present on the shallower 30-meter transect and in fact only selected for habitat categories on that transect. So in this case, the close proximity of the kelp forest and the often productive waters that come with a site on the edge of a canyon contribute more to the species' presence than the microhabitat categories that I was looking at. Okay, some takeaways. These type of characterization studies are important aspects of adaptive ecosystem-based management, particularly in areas or depths that are understudied. Community population dynamics are highly complex, and they depend on a variety of factors, including both seasonality and shifting environmental variables, as well as available habitat. And finally, although this is a pretty unique site, some of the findings highlight the need to take a broader look at this depth range and continue data collection that will be helpful in the management of these precious resources. On that note, I think there's a lot of opportunity to take these kind of studies a bit further. Additional temporal and species assemblage and environmental variables are necessary for a more comprehensive approach to this kind of study. I only analyzed 1 year of data, and while dives were attempted on a weekly basis throughout the year, longer-term, multi-year data would allow for a more comprehensive understanding of the system. 2017 was still considered to have waters hanging around from the 2014-2015 El Nino event, 2014-2015 marine heat wave, and 2016 El Nino event. So waters likely had not recovered from sustained high temperatures and lower upwelling conditions during the period of this study. I also think exploring a wider range of depths would have been helpful in the study. Adding in a transect at the kelp forest ecotone would provide additional insight into species that inhabit the deeper end of the photic zone and potentially lead to more evidence to the question of differences in species composition as a function of depth. I also think that it would be helpful to explore a narrower range of habitat categories to better understand the specific microhabitat category selection of individual species. Okay, I would like to start off by thanking my committee. Scott, you have been the best advisor, and I would not have made it through this program without you. Gita, thank you for your help. Where are you? To see you. Gita, thank you for your help with writing and statistics, your general advice on grad school and life. Thank you for your flexibility and understanding when things were a little tough. And thank you for keeping your door open when I needed to talk. James, thank you for agreeing to take me on as a student and introducing me to the concept for this project. And thank you for doing the vast majority of the camera work on these dives. Andrew, one of the better experiences I had while here at Moss Landing was working with the research team at the Monterey Bay National Marine Sanctuary Office. You and everyone there always made me feel welcome and gave me lots of opportunities to contribute to the research team, including diving and getting the opportunity to work on the Nautilus as a science chair, which was really cool. It's one of the greatest experiences. So back when I started, it was pre-pandemic, and I started as a NOAA Fellow through the Educational Partnership Program, Center for Coastal and Marine Ecosystems. So I would like to thank them for supporting the first few years of this project, and Moss Landing Marine Labs for awarding me the Scholar Award. In terms of people, I need to give a big shout out to the crew of Marine Ops, both current and past. I never worked at Marine Ops, but I spent a lot of time there, and specifically to JD, who couldn't be here today, but I will always appreciate his support as a captain, both on research cruises for classes and when I was a TA for subtitle ecology, and his support whenever I really just needed to talk as well. Jackson and Jenny were both staff at Marine Ops, and they made every day that I had to fill tanks, set up inflatable boats, clean gear, whatever I needed to do that day, much more fun. Thank you to Brian and Diana as well. Oh, there's so many people. Tara, you are so important to Moss Landing, and I hope you know that. If there was anything I didn't know how to do, what to do, I would just ask Tara. And when things were really tough and ever felt awkward, coming to find you in your office. You are a good friend as well as a vital resource. Thank you to the rest of the Moss Landing community, Ivano, Jocelyn, Katie, the rest of the Moss Landing faculty and staff, everyone who keeps the doors open and keeps this place running. There are so many more people listed here, and I could tell a lot of individual stories about all of them, but I will run out of time. I do want to say a big thank you to Michelle Marafini, who was a student here several years back. She's the postdoc researcher on the Surf Zone project, and I wouldn't have been able to get through a lot of my data analysis without her. And my current boss, Jenny Dugan, who is the lead PI for the Surf Zone project at UCSB, has been very supportive while I get through this. But there are many more names up here and many more people that I could be forgetting, so if your name is not up here, please know that I'm grateful. You thought you were only getting short, thank you. I gave Scott his own slide because I really wouldn't have made it to this day without him. There were a lot of reasons this program took as long as it did for me, but he never gave up on me. So he kept me employed through TAing the subtitle ecology class, working on the Surf Zone project. He's always willing to talk, whether that be about life, whatever I'm working on. You read countless versions of this thesis, and you really helped me pick up the pieces of it after the world kind of shut down. And took on much more responsibility as my advisor than he had originally agreed to, even though he's on everyone's committee, and you might have a problem saying no. Yeah. I was able to travel with him in both Baja and Chile for some pretty awesome classes that Moss offers, or offered in terms of Chile. He puts up with my sending fish pictures for identification to this day, all the time. So thank you. I really appreciate you. Speaking of the Surf Zone project, I wanted to really thank you for getting me more involved in this project. I still work on this project, but down at UCSB as a lead technician, and it's just another reason I wouldn't be where I am without you. And so I could include some of these silly photos of a really cool project. I want to thank my family. I'm going to make it through this. For supporting me through this entire journey, even though mostly they just probably couldn't understand why I hadn't graduated yet. And thank you for making the trip up here to watch me as I finally did it. Mom, you are the strongest person that I know. You have shown me what both strength and compassion look like, and given everything you've been through in the last several years, I'm really happy you're here for this. Dad, thank you for your love and support. Kristen, thank you for always being on the other end of the phone line. You have been my friend, my travel buddy, my sometimes roommate, field work helper on occasion. And I really appreciate your support, even when times were low. There's one guy stuck with me through all of the highs and lows. Ish, I can't thank you enough for standing by my side through thick and thin, moving up here for this program, moving back to Santa Barbara, even when you really loved Santa Cruz. Being my sounding board, my constant friend, and my partner through everything. And of course, a shout out to this little black cat that we named Rosemary that I found outside my house in Santa Cruz deep in the pandemic, who carries a heavy load as my emotional support cat. Okay, last one. My friends are like my family, and some of these people are basically my family, including the ones that showed up here today. It really does take a community, and this is an awesome community full of people who I can always call on no matter how far apart we live, and people who will take ocean dips and go dancing and listen to me while I cry or scream or laugh. So I couldn't have done this without them as well. They're all a really cool group of people. So, and with that, I will take any questions.
All right, excellent job, Lauren. Okay, time for questions. And when you ask a question, you have to speak into this microphone because people online will hear you that way. So who wants to ask Lauren a question? Come on, make her squirm a little bit. All right,
Don't
Nate, going again. First question. Out of the gate. Something about statistics, maybe.
Why would you do that?
Great job, Lauren. So in your introduction and conclusions, you mentioned the importance of body size. In the introduction, you talked about ontogenetic shifts in habitat use, and your stereo camera rig looks like it can do some cool measuring of body length, but you didn't discuss that much in the presentation of, on the results side. So I was curious if you had looked at the any differences in the size frequency distributions as a function of those variables you were interested in.
So we didn't look, mostly the size capabilities of event measure we used to calculate biomass. So one idea we had was to look at the size distributions. We didn't end up doing that. I think that would be, it was part of the large scope of this project that I was trying to narrow down. So I think that would be really interesting, especially because there's these ontogenetic habitat usage of most of these species do that. So I didn't look at them. I think that would be really interesting, though.
Thank you.
One thing you did see probably, right, is that there were big density changes with season, but not big biomass changes, because that's all these little tiny little baby fishes showed up and really changed the patterns. Who else? Question?
Hi, Lauren. Thank you so much. That was fantastic. I was wondering, how did you account for fish that swam out of your frame and then maybe back in ones that you saw a few times?
So this was kind of modeled, the stereo rig was flown continuously straight. So if the fish would swim out of the frame, there weren't a lot of fish swimming out and back in of the frame. That happens a lot in like kelp forest settings, and so we have to like be careful. But for this, if they were in the frame at one point, then we would count them as one fish, and then if they swam out and then swam back in, you know, if they showed up separately in the frame, then they would be counted as a fish. But there weren't a lot of kind of double counting. There's a lot of slow-moving species down there that are kind of hanging out in big schools. But
Thank you.
yeah. There's only one way to go into the canyon.
Hi, Lauren.
Hi.
Good job. I have a question because there are a lot of sampling organizations around Monterey Bay, around California coast that sample maybe once, maybe twice a year, and
Yeah.
you sampled multiple seasons. And so given your results, and it was inside a reserve,
Yeah.
right? And a lot of these sample inside and outside. What would you recommend as for the season, or should they modify their sampling to pick up more of the changes you saw?
I think that it would be interesting if you want to look. I mean, these species are often highly seasonal, as we saw. So I think that modifying the data collection to be more frequent throughout the year would give you different results. I know a lot of these organizations, especially even the ones I've worked for too, usually get a summertime community because that's when conditions allow the most diving. So we get like a really good picture of what's happening in the summer. I think this study kind of gives a little bit of evidence to the idea that it would be helpful to sample at different times throughout the year, obviously weather allowing. But yeah.
Hi, Lauren. Great
Hi.
job. That was awesome. I had a question, kind of very specific, about like kelp greenling.
Real fun.
And I know you said like some of your hypotheses or whatever were like based on the males kind of selecting the sites and then the females coming in.
Mm-hmm.
Did you incorporate sex into any of your stats, since you can visually tell if they are male or female?
Right. So for this was more of for kelp greenling, it was a question that I had. I didn't incorporate them into the statistics, but I did have the information on the video files. I could see if they were male or female. So that was more of like a visual observation. But I think that given more data collection, so you can have more numbers and make sure that you can, they are one unique species where you can tell the difference between males and females. Incorporating that it'll lend a lot more evidence to that point, but I didn't incorporate them into my stats.
So anecdotally, did you see more males in the
Yeah.
winter and spring and not as many females?
I did.
All right, we have an online question
Oh, fun.
from your good friend Zoe.
How dare she?
How
She's on
dare
her
she.
honeymoon.
She asked you a really hard question, though. What is your favorite fish, and why?
My favorite fish that I saw on this transect, or like my favorite fish in the world?
She didn't specify, so you could answer this however you please.
Oh my goodness. I think I'll go, my favorite fish that I saw on this transect is my favorite rockfish, which is a rosy rockfish.
Good choice.
They're the cutest. They are pink and orange, and my cat's named Rose. So, that's my answer.
Good answer. Good answer. Any other questions? Oh, there's two more.
Great job. It was great to see all your data come together. You have a lot of data and a lot of analyses, and it's complicated, and I think you did a great job of explaining it. Nate stole my initial question, so I'm going to piggyback off of it. I know you didn't do quite separations between age classes, but anecdotally thinking about it, if you remove, let's say, all the young a year, you're always very smalls,
Yeah.
and only looked at adults,
Yeah.
and removed everything else from the analysis, how do you think those assemblages would change throughout the seasons, only looking at adults that would tend to just stay in that area versus juveniles coming in and moving or shifting?
So most of the species that we saw were adults for, There were at times where I identified like a juvenile, especially because sometimes, as you can see in the images, it's hard to see, but you could say that they were like a yoi, basically, a young of the year. And so for a lot of the analyses, I removed just the yois so that I could look at kind of adult presence only, and that was the majority of what I saw anyways. I wasn't seeing clouds of like tiny, tiny ones. The biggest differences that I saw were these pulses of adults or subadult, the square spot and half-banded rockfish, which are small-bodied species anyway, but they weren't necessarily juveniles. So I think we did do that for this analysis. I removed any of the like odd little yoi species that I found. So these patterns are pretty consistent with just looking at adults.
And I have a second question.
Okay.
You sampled weekly, or tried to sample weekly,
Attempted, yes.
but you binned everything by season.
Yeah, I looked at month as well. We started off binning things by the month, and then the patterns were a little bit more clear seasonally. So they were binned by season, by month and by season, and the seasonal analyses are the ones that we stuck with.
Okay.
Yeah.
So that answers my question. Like that 1 week to the next looked fairly similar to one another. I'm just kind of curious if like you sampled 1 week, you went back the next week, and you saw like a vastly different community, or was it sounds like it was very subtle changes.
It was pretty subtle changes through them, and so when we grouped them into the season, you could see these big differences. And I had all of the monthly data as well, and it was a lot. It was a lot of data, and so we kind of shrank it down to look at the season. And I did see these like seasonal changes that were relatively gradual, except for there was a marked increase in like the fall and winter. But yeah.
And on seasons.
Yeah.
Sorry, one last one. Monterey, I know.
Take the microphone.
It's my mic now. Monterey is known to have kind of odd seasons.
Yeah.
The bay doesn't follow a typical seasonality. And
Right.
It's usually shifted by like a month or two.
Yeah.
And so your groupings of seasons, was that our typical seasonal cycle, or was that the Monterey Bay seasonal cycle where spring starts a little bit later? I think technically spring is starting February, but Monterey spring is like April, May.
Kind of an in-between. My spring started in March.
Okay.
So the spring, and that had a lot to do with the seasonality of the area, but also just the availability of the data that we had. We weren't able to sample in February. So we kind of started with March and then went through December and divided the seasons up like that. So even the winter season is like a little bit more into the fall as well. So.
Okay.
One final question, because Ella had her hand. We gotta finish with a gaucho.
Awesome talk. Kind of to piggyback a little bit on seasonality, I was wondering was it just February or another month that you weren't able to go out?
January.
January. Do you expect that you would see anything different given that you were able to go out during that month? And did you look at like any sensors or instruments deployed during the month to see and kind of group together, well, given this many nutrients that are out or this event, we expect to see these fish be abundant this month?
I looked at the sensor data for those couple of months, but I didn't really pull it. I just kind of looked at it. I wasn't really trying to extrapolate into other months. I think that would be interesting to do, but no, I did not for this study.
Thank you.
You've seen a lot of whitewater.
Yeah.
All right, well, let's give Lauren a big round of applause. All right, her committee is going to whisk her away for a few minutes, ask a few more questions, and then we'll bring her back. But go ahead and start the celebration, and we will join you all shortly.